know its functions. Very, very important. So please, let's use this time. Let's use this time to be attentive for the 2 hours and get something out of it. So it's purely revision. And we're going to go through the whole structure of the nephron, the function of the nephron being majorly osmoregulation and excretion. So of course, osmoregulation is part of homeostasis. And in the topic excretion and homeostasis, we do discuss excretion, how substances are removed from the body which are not necessary in the body. And osmoregulation is basically, or homeostasis is the maintenance of the internal environment to a constant. And osmoregulation being part of homeostasis is maintenance of amount of water and salt in the body. So that is where you're going to, that's what we're going to do now. Okay. Kidneys are major organs in the body. There are two found the lower part of the abdomen. You get that they are usually in pairs. An individual can still survive with one kidney. Meaning that if one is done, a person can still survive with a one kidney, or if one decides to donate one, he can still survive with a kidney. So I've already mentioned the kidney is an organ that is used for excretion. And the substances that are excreted by the kidney is majorly the nitrogenous waste, which is urea, excess water, and salts. Wastes are not allowed to accumulate in the body because they will affect the normal functioning of the body. If they're left to higher toxic levels, then they can. So the major reason why those are removed from the body is to cause the normal functioning of other processes in the body. So urea is usually produced in the liver. Can somebody to describe how urea is formed in the liver? To describe how urea is formed in the liver. Urea is formed in the liver. So can somebody just describe how it is formed in the liver? Hands up, or you can do it in the chat box. How is urea formed in the liver? What is the process of formation of urea?
Deamination.
That is deamination, correct? So describe the process of deamination. Wesley Ondu.
Uh, help.
Describe the process of germination. Wesley, umepotelea wapi?
So the proteins, the excess proteins are taken to the liver, where they are converted into their amino form, the nitrogen and hydrogen. The nitrogen part and the hydrogen part is separated. The nitrogen part is converted to urea in the liver, and it is
This one. This.
Sheldon Wandati, please help.
Formation of urea occurs primarily in the liver. We have the urea cycle. It's also known as the ornithine cycle. It's a crucial metabolic pathway that takes place in both the mitochondria and cytoplasm of the liver. Primary purpose is to detoxify ammonia, which is produced during the breakdown of amino acids and other nitrogen containing compounds. So we have the mitochondrial phase. In the mitochondria, ammonia combines with adenosine triphosphate and carbon dioxide. The reaction involves the enzyme, what is it called? Carbonyl. I don't remember it correctly. So we have the ornithine, a compound which is essential for the cycle and reacts with carbonyl phosphate to produce tryptophan.
All right. Let me. I can get so many answers from the chat. Here we have Peter Njogu. The urea cycle or ornithine cycle converts excess ammonia into urea in the mitochondria of the liver cells. Excess, that is Lorraine. Yeah, okay. Excess amino acids are broken down in the liver through the removal of an amine group, which is reacted with the hydrogen to form highly toxic ammonia. Ammonia is reacted with carbon dioxide in the ornithine cycle in the presence of arginase enzyme to form urea, which is less toxic. That is correct. Excess amino acids, that's Ryan. Excess amino acids are broken down to amino group. Amino group combines with the hydrogen atoms to form ammonia. Ammonia goes through the ornithine reaction where it is converted to less toxic urea. All right. Christian already here is the breakdown of excess amino acids through the removal of amino group, which is reacted with the hydrogen to form toxic ammonia. Toxic ammonia is reacted with the carbon dioxide in the ornithine cycle in presence of arginase enzyme, hence forming less toxic urea. The urea is then taken to the kidney for excretion. The water is used as metabolic water, and carboxylic group is converted to skeletal carbon, which is oxidized. That is very correct. Already, that is excellent. First of all, we start with the excess amino acids. Yeah, we don't talk of proteins being taken to the liver. Proteins are digested to amino acids in the digestive system. They are converted to amino acids. So the amino acids are taken to the liver through the hepatic portal vein. Now, it is only amino acids which are in excess. If the amino acids are not in excess, they are not converted to urea. So if you feed on large amounts of proteins, and then all of them converted to amino acids, the body will only pick a little amount of amino acids that is necessary. The rest are converted to urea, which is removed from the body. So that's why you're advised to eat little amount of protein or just enough, yeah, because you'll only be overworking the liver to form urea. So in this case, it is excess amino acids which are in excess, which are broken down by the liver cells. The amino acids usually have the amine group or the amino group. That's the NH2. The NH2 group is converted to ammonia. Ammonia is highly toxic and therefore cannot be left to accumulate in the body or in the liver. So what usually happens, the ammonia reacts with the carbon dioxide in the ornithine cycle. And that is why the adigenase enzyme is present, usually to speed up that process. So when the ammonia reacts with the carbon dioxide, it forms urea, which is less toxic. That urea is usually now taken to the kidney for excretion. So from the amino acid structure, we have the amino group, we have the carboxyl group, and we have the hydrogen carbon chain, the hydrocarbon chain. So the carboxyl group is what is converted to carboxyl group together with the hydrocarbon chain is what is oxidized to form energy, or it can still be used to form glucose. And so, of course, we've already removed the amino group, and now we're remaining with the carboxyl group and the hydrocarbon chain. So that is now that is how deamination occurs. Yeah. So we're only describing how urea is formed. How urea is formed. And now later on, we'll be looking at the kidney, how the urea is removed from the kidney. Yeah. So, okay, some are saying, I repeat, the process of deamination. For those who have not understood, I've said after the process of digestion of proteins, amino acids are taken to the liver. The proteins are usually digested to amino acids. Amino acids are the end products of proteins. They are absorbed in the ileum and they are usually taken to the liver. The purpose of the liver, why food, all the digested food substances have to go through the liver, are majorly two reasons: for regulation and for detoxification. So in this case, we are also actually talking of regulation of amino acids. So when the amino acids are in excess, all the amino acids have been absorbed from the ileum and they are taken to the liver. If they are in excess, they will not be let to be distributed into the body by the blood. The excess will be deaminated. So the description of deamination or the process of deamination is basically removal of the amine group from the amino acid. The amino acid structure is made up of the hydrocarbon chain, the carboxyl group, and the amino group. So from the amino acid structure, the amine group or the amino group is what is usually removed from in the process of deamination. The amine group is NH2. That is the formula for amine group, NH2. The NH2 is usually converted to ammonia, which is NH3. Ammonia is highly toxic in the body and cannot be left to stay in the liver. So it is usually converted to urea. Now, how is it converted to urea? It is usually converted in the process referred to as the ornithine cycle. In this process, the ammonia reacts with the carbon dioxide to form now urea. Urea is a less toxic substance which is removed and taken to the kidney through the blood. So of course now from the liver, you can describe the process, how that urea will move from the liver to the kidney in the circulatory system. So can somebody just describe how that urea will get out of describe the pathway, how the urea will get out of the liver until when it gets to the kidney. Now the urea has been formed. Yeah, urea has been formed in the liver. How will it get to the kidney? Describe the pathway. Describe the pathway. Can I get participation? Hands up. Describe the pathway of how urea, which has been formed in the liver, gets to the kidney for excretion. So this is now circulatory system. Hands up, or can I see the charts? Pathway of movement of urea from the liver to the kidney. Tuongee vijana. Who's ready to answer that? Let's participate. All right, from the charts, I can see this is good. So it will, here Jason Mwangi says, it will go out of the liver through the hepatic vein into the heart through the vena cava, and out of the heart through the aorta, and into the kidney through the renal artery. So you should also describe how it gets out of the heart to the lungs and all that. That is the pathway. The kidney receives blood that is rich in nitrogenous waste, such as from the liver through that is Victor Kamau. I want you to describe the pathway where, how will this urea get out of the liver until when it is excreted in the kidney, until when it gets to the kidney. What Mark here says, the urea enters the bloodstream via hepatic vein, correct? And pumped to the heart. It is not pumped, it is taken to the heart. So he says the urea enters the bloodstream via the hepatic vein and pumped to the heart for oxygenation of blood. Oxygenation does not occur in the heart; it occurs in the lungs. Blood, after which it is pumped out of the left ventricle via the aorta, which branches to the renal artery that transports blood to the kidney under high pressure. There's something, yes, correct, but there's a lot that is missing there. Hugh says urea is released from the liver cells to the bloodstream and is transported to the kidney. Now, from the bloodstream to the kidney is what I'm looking for. It is released from the liver cells to the bloodstream, yes. How does it get to the kidney? Here, Justin Busolo says the urea is released into the hepatic vein where it is passed into the posterior vena cava. Is it posterior? It is actually inferior vena cava. We have inferior and superior. So it is passed into the inferior vena cava where it goes through the pulmonary artery, taken to the heart for oxidation. Oxidation does not occur in the heart, the lungs. And oxidation, it is oxygenation. Taken to the heart, so it is usually taken to the heart, not for oxidation, not even for oxygenation. And then, so it says, taken to the heart for oxidation of the blood and brought back into the heart by the pulmonary vein. It then flows into the aorta, where it flows into the dorsal aorta, and then flows into the renal artery, into the kidney, ultrafiltered by the Bowman's capsule, into the renal tubule. So I've just mentioned it's not taken to the heart for oxygenation. Oxygenation occurs in the lungs. Already says from the liver, urea is transported to the general circulation through the hepatic vein, correct? Transported to the heart through the vena cava, correct? Transported to the lungs through the pulmonary artery, that is right, for oxygenation, correct? And back to the heart through the pulmonary vein, correct? It is transported out of the heart through the aorta and transported into the kidneys through the renal artery. That is correct. So that is it. Hepatic vein, inferior vena cava, right atrium, right ventricle, pulmonary vein, lung, pulmonary artery, left atrium, left ventricle, aorta, then renal artery. So that is the right pathway. So now this urea has now been taken to the liver. To the kidneys. Now we look at the structure of the kidney, and that's how it looks like in your screen. That is a little complex. Let me share you the simplest structure of the kidney So the simplest structure of the kidney, when you look at it, this is it, made up of the cortex, it's made up of the medulla, and it's made up of the pelvis. And you can see even the colors there indicate exactly how they look like. Yeah. And we get that the kidney is supplied by the renal artery. The renal vein is the one that drains blood out of the kidney. So basically that's the structure of the kidney. As simple as that. Now, when you look at those shapes of those, or the sizes of those parts of the kidney, the three parts, that's the cortex, the medulla, and the pelvis, they're almost similar in size with this particular diagram that you're able to see. But later on, we'll see that in some organisms or some animals, the structure of the cortex may be wider or narrower. The structure of the medulla may be wider or narrower, depending on the adaptive features of this particular organism or where it stays. So we'll look at that later on. So that is the kidney. Now, the structure of the nephron. When you look at the structure of the nephron, or before we look at the structure of the nephron, we'll want to look at which part of the nephron is found where. But let's just look at the structure of the nephron and see what is it made up of. The nephron is made up of, you can see, made up of the glomerulus. It's made up of the proximal convoluted tubule. It's made up of the loop of Henle, descending and ascending. And then it's also made up of the collecting tubule. Majorly, distal convoluted tubule and then the collecting tubule. So those are the major structures that are found that make up, those are the structures that make up the nephron. Now, when you look at the structure of how the part of the nephron, which part of the nephron is found in which part of the kidney? And you look at this diagram here, you can see that the cortex there usually contains majorly the Bowman's capsule, proximal convoluted tubule, and distal convoluted tubule, while the medulla is made up of the loop of Henle. The loop of Henle is usually contained within the medulla. So, having that knowledge of what is found where, which part of the nephron is found, which part of the nephron, where each part of the nephron is found in the structure of the kidney. Medulla and cortex is part of the kidney. You can see that the renal pelvis there is basically the collecting duct. So, adaptive features, I will bring this later on, the adaptive features of a certain animal to a given environment, depending on where it stays. It's also dependent on how the size of the kidney appears, the size of part of the kidney appear. So having the knowledge that it is usually the proximal convoluted tubule and the Bowman's capsule that is found in the cortex, and the medulla usually majorly contains the loop of Henle, then we will see why would, for example, the cortex of the kidney of a given organism be narrower or wider, and why would the medulla of the kidney of a given organism be wider or even. So we will look at that much later. For now, let's look at the nephron. So that is the structure of the nephron. The reason why the nephron is found in the kidneys is basically it's the basic unit of excretion in the kidney. We have several millions of nephrons within the kidney, and they play a major role of separating urea and other wastes from the body, separating urea from other wastes from the body. We've seen how urea is formed. We've seen how urea is taken to the kidney. So as it gets to the kidneys, it is usually the renal artery that will take this blood full of urea into the kidney. So the reason why urea is highly concentrated in the renal artery is for the purpose of excretion. So you find that renal artery and renal veins are some of the unique blood vessels inside the body, apart from the pulmonary artery, pulmonary vein, and the hepatic portal vein. Why is it special? Is because many arteries or majorly arteries carry blood with a larger quantities of end products of digestion and little amount of nitrogenous waste. For the renal artery, it is unique because it is an artery and it contains larger quantities of nitrogenous wastes. The renal vein, on the other hand, is also unique because veins, what you know about veins, they carry blood with a lot of carbon dioxide and more of wastes. The renal vein, in this case, will contain little amount of it will contain little amount of nitrogenous waste because most of it would have been removed by the kidney. Now, we're going to look at each and every part. It is being supplied by blood, oxygenated blood with a lot of oxygen by the renal artery. It branches renal arteriole, which branches further to the afferent arteriole. Which branches further to the afferent arteriole. The afferent arteriole branches into smaller blood vessels, which are the capillaries. The capillaries form what is referred to as glomerulus. The glomerulus are very tiny, they are tinier compared to the afferent arteriole. Then later on, they coalesce to form the efferent arteriole. This efferent arteriole is the one that ramifies the whole of the nephron, as you can see from that diagram. Now, there's a process called ultrafiltration that occurs in the glomerulus. Ultrafiltration is basically the forcing out of substances from the blood within the glomerulus into the Bowman's capsule. You can see the structure of the Bowman's capsule. It's adapted in such a way that it's cup-shaped, or funnel-shaped, cup-shaped to contain the glomerulus. Yeah. And also to receive the glomerular filtrate substance to get into now the proximal convoluted tubule. Now, what enables ultrafiltration? What enables ultrafiltration to occur in the glomerulus? What enables ultrafiltration to occur in the glomerulus? Adrian Degwa says it's high blood pressure. Where? High blood pressure from the renal artery. Cyril Obiambo says it's pressure. Describe how this high blood pressure causes ultrafiltration. Jason Mwangi says the existence of pores on the capillary walls and wall of the Bowman's capsule. Ryan Tuku says the difference in the lumen of the afferent arterial and the efferent arterial, correct? Abdullahi, high pressure which causes passage of small particles while large particles are filtered. And then Kevin Kipchumba, they say the lumen of the afferent arterial is greater, is wider. We expect it to say, mention the lumen is wider than the efferent arterial, that's creating high pressure, correct? Yes. So basically, we can mention about Joseph Karanja says high blood pressure because of different thickness of afferent and the efferent arterioles. Yes, most of you are mentioning the afferent and the efferent arterioles. Correct. So it's true that the structure of the afferent arteriole, the size of the lumen of the afferent arteriole is wider. Afferent arteriole's lumen is wider compared to the size of the lumen of the efferent arteriole. So that is one. Another thing is that there's blood in the renal artery, and the renal arteriole comes in at high pressure. Yeah. So once it gets into the afferent arteriole, now which branches further now to tinier blood vessels. Yeah. There is the reduction in the size of the glomerulus is what makes ultrafiltration to occur. So we get that the blood or the blood vessels are not able to resist that pressure. And therefore it causes the oozing out of the liquid part of the blood out. Yeah. So when you look at the sizes, I just want to share. I want to share the size of the afferent arteriole and the size of the efferent. Yeah. So when you look at this particular diagram, the vessel through which the arrow is pointing inwardly is the afferent arteriole. Yeah. And the vessel through which the arrow is pointing outwards is the efferent arteriole. Yes. So you can compare the two sizes. So as blood gets in through the afferent arteriole with quite high pressure, it branches into smaller blood vessels there, which are the blood capillaries, and the blood is leaving through that afferent arteriole, which is now has a tiny lumen, compared to the afferent arteriole. Pressure will be built up. That will cause the pressure to be built up in the glomeruli, causing blood substances within the blood to ooze out or to be filtered out. And that is what we refer to ultrafiltration. So here is just to show you the comparison of the size of the afferent arteriole and that of the efferent arteriole. Now, what are the substances that are filtered out in the glomerulus? All right, somebody's telling me to slow down. Okay, I will slow down a bit. What substances are filtered out of the glomerulus? Abdullah says the blood plasma proteins. What substances are filtered out? To form the glomerular filtrate, because once it is filtered out, it gets to the Bowman's capsule. Ultrafiltration. So somebody says all nutrients, metabolic waste, mineral salts, vitamins, amino acids, water and urea, metabolic wastes, glucose, blood cells,
Acids, vitamins, urea, sodium chloride, water, sodium and vitamin.
Yes, glucose, amino acids, vitamins,
Urea,
urea.
sodium chloride, water,
yes,
sodium hydrogen carbon.
So mineral salts, urea, and the end products of digestion like glucose, amino acids, vitamins, and of course water. Yeah. So once you know what substances will get out to form the glomerular filtrate, yeah, the key reason why ultrafiltration occurs is for the removal of urea. So urea is a key substance that is filtered out. We usually have water. Any substance with smaller sized molecules is filtered out. And this includes glucose, amino acids, vitamins, mineral salts, water, and urea. Yes. So Jeremy there puts it clearly, small size substances, e.g. water, urea, glucose, mineral salts, and amino acids. What is usually not filtered out from the blood? What is not filtered out?
Blood cells and plasma proteins.
Yes, the blood cells and the plasma proteins. And the reason why they are not filtered out is because they are larger particles which cannot fit into the pores of the capillaries. Yes. So Now, our concentration is what has been filtered out.
Mm-hmm. Yeah.
The substances that have been filtered out is what you're going to look at. You're going to look at the urea, you're going to look at water, you're going to look at glucose, amino acids, salts, and vitamins. These substances, they are those which are very useful. Yeah. There are substances which are useful, and therefore they have to be taken back to the body. Now, we look at now the structure of the nephron. Let's go back to that structure of the nephron. In this structure of the nephron, we have the substances that are filtered out, which will be reabsorbed back to the bloodstream at different portions. From the Bowman's capsule, the glomerular filtrate, whatever that has been filtered out is called glomerular filtrate. The glomerular filtrate will get into the proximal convoluted tubule. So, the proximal convoluted tubule, you can see when you look at it, it is highly coiled. That's the modification of the proximal convoluted tubule.
Madam, it's convoluted.
Yes. The structure of the proximal convoluted tubule is highly coiled one. And the reason why it is highly coiled is to ensure that the substance or the glomerular filtrate moves slowly within it to cause more reabsorption. Yeah. It reduces the speed of flow of the filtrate and therefore causing more time for more reabsorption. The word there is reabsorption, not absorption. Why it is reabsorption? Why it is reabsorption is because they were initially absorbed. Those are substances which were initially absorbed into the bloodstream in the alimentary canal. So right now, they are reabsorbed again, or they are being absorbed again into the bloodstream at the liver or at the nephron. So the highly coiled structure there is to slow down the speed of the flow of the filtrate to cause more reabsorption. The cells lining the tubule, the proximal convoluted tubule, has numerous mitochondria, yeah, to provide more energy for active reabsorption. There's active transport that occurs there. So, in the process of active transport, you expect that energy will be required, and therefore energy can only be provided by mitochondria. So, how do you put that statement? Is the cells lining the tubule have numerous mitochondria. You will not say that the tubule has numerous mitochondria or the tubule has or contains a lot of mitochondria. Please get it clearly. That's how some of you fail. It is the cells lining the tubule. The mitochondria are usually found in the cells. Mitochondria are organelles. So they are found within the cells that are lining the tubule. The cells, another adaptive feature is that the cells also lining the tubule have a lot of microvilli. Not villi, just microvilli. Yeah. And this increases surface area for reabsorption. The tubule itself is quite long to provide a larger surface area for reabsorption. So get the difference between length and coiling. Yeah. The coiling there is to reduce the disk speed, while the length is to provide a larger surface. So whatever that provides larger surface is the length and the existence of the microvilli. And then it is supplied with blood capillaries for transportation of absorbed substances.
Pardon.
So I've said the adaptive features of the proximal convoluted tubule. Yeah. So when you look at the adaptive features, highly coiled to slow down the movement of the glomerular filtrate within it to cause more reabsorption. It is long to increase surface area for reabsorption. The cells lining the tubule have microvilli. The microvilli there is to increase surface area. So you can see of surface area is by the length of the tubule and the existence of the microvilli. Yeah. So I was insisting that please use proper description. Yeah. For example, you say the cells lining the tubule have microvilli. Do not say that microvilli, the tubule contains microvilli. You will not say that the tubule contains microvilli, but you'll say that the cells lining the tubule have microvilli to increase surface area for absorption. And then you also say the cells lining the tubule have numerous mitochondrial active uptake of the cell. Why? About the coiling of the filtrate flows. So the speed is reduced. So those are the adaptive features of the proximal convoluted tubule. Now we look at what is reabsorbed back into the bloodstream at the proximal convoluted tubule.
Madam Emai, this is Ondiro here. Yeah,
yes.
we do a roll call quickly first before you continue.
Yes.
Just pause there a little. Boys, you look at the screen, pick your class, and submit. Let's go roll call. This will take us about 1 minute or so. Quick roll call. Roll call. We should have finished now. I think this is 3C. So 3K, you're doing very well. 4Q, we still want you. We can't see you. 4Q, this number was very, very small.
4Q. We have only 4 from 4Q.
We still have some who have not done the roll call. We have about 170 and only about 125 have done the roll call.
Please finish up with the roll call.
Okay, we are ending the roll call now.
Almost. You still have 25. You still have 25 who have not done the roll call. Ah, all right.
That is it. We can continue. Thank you, guys. Madam Emai, you may proceed now.
All right, so. Okay, we can continue. So your participation is quite low. It's not good. Now, let's continue from the proximal convoluted tubule. We are talking of the substances being reabsorbed. In this case, there are specific substances that are reabsorbed at the proximal convoluted tubule. And these are the substances. We have all the glucose. All glucose, and you can see I'm saying all glucose, not just glucose. Neither am I saying some glucose. All glucose, all amino acids. So those two substances, all of them are reabsorbed at the proximal convoluted tubule. All glucose and all amino acids are reabsorbed at the proximal convoluted tubule. That is why the urine of a normal person does not have glucose. Urine of a normal person doesn't have amino acids because all of it has been reabsorbed at the proximal convoluted tubule. We have some water. Yes, Kendrick, you have a question?
Yeah, please, before you finish, can you please explain how the release of less or no insulin connects to resulting to diabetes mellitus?
Let's answer that by the end of the lesson because now we'll be deviating from what you're doing. I will answer that by the end of the lesson. So,
Okay.
yeah. Now, So we've said all glucose, all amino acids are reabsorbed at the proximal tubule. We also have water, the mineral salts, the PCT. So know what's reabsorbed, because each and every part of the nephron has its specific function. Please know that. Each and every part has its own specific function. And that's why we're now looking at the role of the function of the nephron. The role of the Bowman's capsule is to contain the glomerulus, and also for ultrafiltration, to cause ultrafiltration, or to contain now the glomerular filtrate that is now getting into the renal tubule. Now that glomerular filtrate, we've said what is contained in the glomerular filtrate. What is contained in the glomerular filtrate is water, urea, amino acids, glucose, vitamins, mineral salts, and salts. So all those substances are not going to form urine. Some are going to form urine, while others will be taken back to the bloodstream because they are useful substances in the body. Now, this taking back of the substance back to the bloodstream is what is referring to as reabsorption. And it is usually referred to as selective reabsorption. Why is it selective? Because not all substances are taken back. It's selectively taken back. Some of the substances will be taken back to the bloodstream, others will not. The key thing why the nephron is there is to filter out the urea from the body. You filter out the urea from the blood to form urine, to be taken out. And when you look at urea itself, it's not just urea. When you look at urine, urine is majorly made up of water. The quantity of urine is measured by the amount of water it has. So we'll find that excess water will be taken out as part of the urine. And that's one of the functions. That's why we say the kidney is an osmoregulatory organ. What is osmoregulation? It's the regulation of the quantities of salts and water. Yeah. It is an excretory organ. Why? Because it's removing nitrogenous waste or wastes, which is basically urea. So osmoregulation in this case, it depends with the osmotic pressure of the blood, of the body, causing either more water to be lost, not to be reabsorbed back to the blood system, or to be reabsorbed. So if less water is reabsorbed, most of it is taken as urine. If more of it is reabsorbed, then less water will be taken out as urine. So that's now osmoregulation. Now it is going to regulate amount of water and also the salts. Osmoregulation and excretion is a key function of what? Of the kidney. So we've looked at what is reabsorbed back at the proximal convoluted tubule. Now, the glomerular filtrate now will get into the loop of Henle. We have the descending loop of Henle and we have the ascending loop of Henle. The two have their own functions also. Now, the glomerular filtrate that will get into the loop of Henle does not have glucose, neither does it have amino acids. In the descending loop of Henle, there's more reabsorption of salts, the sodium chloride ions in the loop of Henle. In the descending loop of Henle, there's more reabsorption of water. In the ascending loop of Henle, there's more reabsorption of sodium chloride ions. So, majorly, the function of the loop of Henle is for absorption of sodium chloride ions that is salts, generally salts, and absorption of water, and absorption of water. A lot of water is reabsorbed back. Reabsorption of water, a lot of water is reabsorbed back at the loop of Henle. Yeah. That one describes sorry, I'll be slow. That one describes now the structure of the sizes of the parts of the kidney. Yeah. Dependent on how much water should be reabsorbed back to the bloodstream will determine the size of the medulla, will determine the size of the medulla of the kidney. Okay. Now, the reabsorption of salts is facilitated by two things. Production of which hormone?
Aldosterone.
Correct. Aldosterone hormone. Where is it produced from?
Adrenal glands.
Adrenal gland.
Adrenal glands, correct. Where do you get the adrenal glands in the body?
Right above the kidneys.
Above his kidney.
Correct. So you get that adrenal glands produce aldosterone hormone. Aldosterone hormone is usually produced when the osmotic pressure of the body is low. When the osmotic pressure of the body is low, it means that the body fluids have more water and less salts. More water, less salts. Dilute. So what usually occurs in the kidney, specifically now at the loop of Henle, there'll be more reabsorption of salts back to the bloodstream. Now, the aldosterone hormone will be produced or will be released to cause more reabsorption of salts back to the bloodstream. If the osmotic pressure is high, meaning there are more salts and less water in the body fluids, then less or no aldosterone hormone will be produced. Less or no aldosterone hormone will be produced. And therefore, it will cause less or no reabsorption of salts back to the bloodstream. In this case now, it will cause more salts to be released as part of urine. So you get that there'll be release of a higher concentrated urine with more salts. If the body fluids have a low osmotic pressure, meaning it has more water and less salts, more salts will be reabsorbed back to the bloodstream. And then causing less water to be reabsorbed, causing the urine to be dilute and be in high quantities. And that is how osmoregulation comes about. Yes, Christian, your hand is up.
I was talking about the aldosterone hormone. So,
You are?
in my opinion about the aldosterone hormone, according to what I know, the aldosterone hormone
it stimulates the pumping of sodium chloride ions. And so, when the sodium chloride ions are in the bloodstream, they increase the osmotic pressure of the blood, resulting in more reabsorption of water. Then when the water is reabsorbed in excess, it dilutes the ions themselves. Hence, this now lowers the pumping of aldosterone, the secretion of aldosterone hormone. So aldosterone hormone is there to increase the reabsorption of water.
Aldosterone, you're saying aldosterone hormone is to increase the reabsorption of water?
Yes.
Aldosterone hormone is used to increase reabsorption of salts. It is the antidiuretic hormone that causes more reabsorption of water. Do not confuse the antidiuretic hormone and the aldosterone hormone.
Yes.
Yeah, get it clear as what I'm describing. Aldosterone is usually produced when there is what? Decreased quantities of salts within the body fluids. So it causes more reabsorption of salts back to the blood system. Yeah. And that's how osmoregulation now comes in.
Yes.
Yeah. Please get it clearly that way. Okay. So I hope I'm clear. Pale penye nimeelezea how aldosterone hormone comes in. Sawa? To do with osmoregulation. That's why you can now see the function of the nephron in osmoregulation. Yeah. When the amount of salt in the body fluid is lower, then it causes more reabsorption of salt back to the bloodstream by the aid of the aldosterone hormone. Yeah. Because we know that salts have been filtered out at the glomeruli. Okay. But when the salts are high, there'll be reduced reabsorption of salts back to the blood system. And most of it now will be removed as part of urine. Okay, so that is now what happens at the loop. So facilitated by the counter current flow and water within the blood capillaries, as well as as well as what? The glomerular filtrates. When you look at that diagram there, within the nephron is the flow of the glomerular filtrate, and you can see the flow of the the flow of blood within the blood capillaries. They are flowing in opposite directions. Flowing in opposite directions. That's what we refer to as counter current flow. Somebody talking? Please mute. Who is this? So that diagram explains well the counter current flow. That one enables more reabsorption of salts back to the bloodstream. Yeah. The movement of blood and the glomerular filtrate in opposite directions enables now the blood to pick up the salts. And this movement of salts is either by diffusion or by active transport. And therefore, you expect that the cells of the nephron, the cells of the loop of Henle also contain a lot of mitochondria for active reabsorption of salts. Yeah. So the salts are absorbed either by diffusion or by active transport. Okay. Now from there, we now get to the distal convoluted tubule. In the distal convoluted tubule, you can see it is also highly coiled, just like the proximal convoluted tubule. So it has features similar to that of the proximal convoluted tubule. What usually occurs in the distal convoluted tubule is reabsorption of water. A lot of reabsorption of water occurs in the distal convoluted tubule, as well as salts also. As well as salts. So, most of the water is reabsorbed at the distal convoluted tubule by the antidiuretic hormone, also called what?
Vasopressin.
Vasopressin. Where is it produced from?
Pituitary gland.
The pituitary gland, correct. So, just like how I described the release of aldosterone hormone, is also how the antidiuretic hormone is released. It depends with osmotic pressure. Yeah? It depends with the osmotic pressure of the body fluids, whether it is high or it is less. Yeah? So if the osmotic pressure is high, more antidiuretic hormone is released. What do I mean by high osmotic pressure? It means that the body fluids are concentrated, meaning there's less water and more salts. So if the osmotic pressure is high, more antidiuretic hormone is released to cause more reabsorption of water back to the blood system. And when the osmotic pressure is low, which means that there's more, the substances or the body fluids are dilute, having more water and less salts, then it causes less or no antidiuretic hormone to be released, causing less reabsorption of water. And all this will have an effect on the type of urine which is produced. The type of urine in this case will either be concentrated and little or a lot and dilute. And that's now what brings about osmoregulation. That brings about osmoregulation. So, antidiuretic hormone, what the antidiuretic hormone does is usually to cause more permeability of the capillaries around that region, and also the distal convoluted tubule to water. Yeah. So once it is released, it causes the walls to be more permeable to water of those structures. So more water is lost, more water is reabsorbed back to the blood system. Yeah. Bringing now the osmotic pressure back to normal. Yeah. Now that is what causes the osmoregulation. Osmoregulation. So, so you have seen how salts are regulated and how water is also regulated. You can see now by the time we get to the distal convoluted tubule, most of the substances have been reabsorbed back. All the necessary substances have been reabsorbed back to the blood system. All glucose, all amino acids, the vitamins, mineral salts, and some water and some salts, depending on the osmotic pressure. The salts and the water are reabsorbed depending on the osmotic pressure of the body. So all those substances have been reabsorbed. The only substance which has not been reabsorbed and which has to be removed is urea. So by the time the glomerular filtrate gets into now the distal convoluted tubule, those are the higher quantities of substance present there is urea, water, and some salts. Now those one now will trickle into the collecting tubule, which will now join the collecting duct to get into the urinary bladder as urine. So once it gets into the collecting tubule, actually that is just urine, because now in the collecting tubule also there's a lot of reabsorption of water. There's the reabsorption of water in the collecting tubule. So reabsorption of water may continue a bit, and then now once it gets into now the urinary bladder, that will now be urine. So you can see the function of the nephron in excretion and in osmoregulation. If you're told describe the nephron or the kidney as an osmoregulatory organ, that is what you talk about. You'll talk specifically to do with reabsorption of salts and reabsorption of water. And the two hormones involved, aldosterone and antidiuretic hormone. Yeah. And osmoregulation in this case will occur to bring a balance of the quantities of salts and water within the body fluids. Now you'll describe if the osmotic pressure is high, and also when the osmotic pressure is low, what happens and which hormone is released from which part, which gland releases that particular hormone. What is the effect of that particular hormone on the nephron? So now coming back to the structure. Yes, somebody, Benjamin, you're asking how I can show diagrams of animals' different habitats. I will only show you how the structure of the parts of the kidney will be modified dependent on dependent on the habitat of that particular organism. Now, with this knowledge that we know, yeah, we've known that when you look at this diagram, you're talking of which structure of the nephron is found where. From this diagram, you can see that the medulla contains a major part of the loop of Henle, majorly. In the cortex, we get that, of course, we have those cortical nephrons, but majorly, what usually contained in the cortex is the part of the nephron that is contained in the cortex is the Bowman's capsule. Now, looking at the kind of organisms and their habitats, we get that there are organisms that are found where there's a lot of water, and the organisms that survive with little amount of water. This one will cause the organism to have different shapes of the nephron. Let's first of all describe an organism that stays in dry areas, which is subjected to little amount of water in its body. In this case, we will talk of this organism, the organism in this case, because it requires more water within its body, and therefore it has to lose less urine, isn't it? So to excrete little amount of urine and to preserve more water or to conserve more water within its body. So if it's supposed to conserve more water within its body, then it has to cause more reabsorption of water back to the bloodstream. The loop of Henle plays a major role in the absorption of water. So we expect that this organism, first of all, from the Bowman's capsule where ultrafiltration takes place. The more ultrafiltrate is formed, the more the urine. If more ultrafiltrate is formed, if more ultrafiltration occurs, or if more glomerular filtrate is formed, then there'll be more release of the urine. Get it clearly that way. More ultrafiltration can only occur if the Bowman's capsule if the glomerulus is large. If the glomerulus is large, a lot of glomerulus within, then there'll be more ultrafiltration. And in this case, to contain more glomerulus, then we expect a larger Bowman's capsule. So for these animals which live in the where there's less water in arid areas, we expect that they should have a smaller Bowman's capsule to contain little amount of glomerulus to cause less ultrafiltration. And therefore, we expect that they will have a smaller sized cortex. The size of the cortex will be small because what does it contain? It contains Bowman's capsule, which are small in size. Why the Bowman's capsule small in size? To contain little glomerulus. Why little glomerulus? To cause little ultrafiltration. Yeah. And then we expect that the loop of Henle of this particular organism, of the kidney, of the nephron of this particular organism is long, quite long, to cause more reabsorption of water. So therefore, we expect that the medulla will be wider, longer loop of Henle. So the organisms, for this case, we expect that organisms that stay in arid areas, we expect that the cortex is small in size, while the medulla is wider in size. And I've given you reasons. Cortex small in size because they contain smaller sized what? Bowman's capsule. The medulla is long in size to contain a longer loop of Henle. This happens with the organisms that live in plenty water or subjected to have a lot of water. For these organisms, we expect that the Bowman's capsules are wider, are larger, to contain larger glomerulus. Yeah? The larger glomerulus in this case will cause more ultrafiltration. So as I told you, if there's more glomerular filtrate formed, then more urine will also pass more urine. Now, so we get that the Bowman's capsule is wider to contain more glomerulus to cause more ultrafiltration. So what do you expect with the size of the cortex? The cortex will be larger. The cortex will be larger to contain a larger Bowman's capsule. Now, because it doesn't require a lot of water in its body, okay, so getting back, thank you for reminding me. The concentration of urine, somebody has told me, the concentration of urine before we get now to the what? To the animal that is subjected to a lot of water or lives within the water or has a lot of water within its body. Going back to the organism that stays in the arid area, having a smaller Bowman's capsule to contain small glomerulus and having a wider or a longer loop of Henle, causing more reabsorption of water back to the blood system. It causes now the urine to be little amount, and also to be concentrated. Yeah. So the urine will be in little amount and be highly concentrated because most of the water has been reabsorbed back to the blood system. Now, getting back to this animal that is will have a lot of water in its body or subjected to maybe a lot of water, will have a larger Bowman's capsule with a larger glomerulus. More ultrafiltration occurs, and therefore you expect the size of the cortex to be wider. For the loop of Henle, you expect it to be shorter because it doesn't require a lot of reabsorption of water back to the blood system. Therefore, the loop of Henle will be shorter, causing less reabsorption of water back to the blood system. And because the loop of Henle is shorter, then you expect that the medulla will also be narrower. So in this kind of organism, the kidney will appear in such a way that the cortex is wider and the medulla is small sized, and therefore less reabsorption of water takes place, more of it will be removed in urine. And the urine of this organism will be large quantities and dilute. Some Mark asks, is it smaller glomerulus or fewer glomerulus? Glomerulus is majorly made up of capillaries. Fewer or what is it say? Fewer or smaller glomerulus? Alvan says both. You can say fewer, smaller, or Terence says smaller glomerulus. As long as you understand what glomerulus is, isn't it? You can talk of, but majorly we are talking of a smaller sized Bowman's capsule. Glomerulus, we can talk in terms of okay. Further research. I'm also now getting confused. Which one is which? Fewer glomerulus, smaller glomerulus.
Teacher, I can answer. Amo,
Yes.
we normally say smaller Bowman's capsule to accommodate fewer glomeruli.
Thank you. Thank you. You said smaller glomerulus to accommodate fewer glomeruli. Thank you, Madam Hamo, for that correction. So, fewer is the right word. Fewer glomerulus. All right, now where were we? You're saying, pardon, we are saying fewer glomerulus smaller sized Bowman's capsule, fewer, not small, not smaller glomerulus, but fewer glomerulus. Okay. Now, so that is the structure of the kidney dependent on the where the organism, depending on the type of organism and where it stays, the habitat, depending on how much water it's subjected to and whether it's less or a lot. Okay, now since a question was asked, effects of the following quantity, effects of the following on the quantity and composition of urine. Drinking large amount of clean water. Number 2 is drinking of very salty water. Number 3 is removal of the pancreas. That is the question Kipruto asks. What is the effect of the following on the quantity and composition of urine? Number 1, drinking large amount of clean water. Can somebody answer that? What is the effect on the quantity and the composition of urine when someone drinks large amount of clean water?
The urine will be less concentrated.
With what?
The urine will be less concentrated with what? Yes.
Production of large amounts of dilute urine.
Production of large amounts of dilute urine. Explain. Explain why there is a production of large amount of dilute urine.
The hypothalamus stimulates the pituitary gland to secrete less antidiuretic hormone, which makes the kidney tubules less permeable to water.
Before you get to the hypothalamus secreting less antidiuretic hormone, what are the signals? Why would the hypothalamus produce less antidiuretic hormone? Any reasoning? Why would the hypothalamus produce less antidiuretic hormone? The question was, explain the effects of the following on the quantities and composition of urine. Drinking large amounts of clean water. Somebody mentioned it causes production of large quantities of dilute urine. And the explanation is it is because the hypothalamus stimulates the pituitary gland to release less antidiuretic hormone. So before you get there, you have to explain why is less antidiuretic hormone produced?
Low osmotic.
Yes. Somebody was answering and you disappeared. Kendrick?
I think there will be production of less antidiuretic hormone as there will be low osmotic pressure already. The blood will have low osmotic pressure already, so there will be no need of absorption of more water, hence production of less antidiuretic hormone.
Yeah, because now drinking a lot of water causes more of it to be absorbed into the blood system, reducing the osmotic pressure. So once the osmotic pressure of the body fluids has reduced, then less antidiuretic hormone will also be released to cause less reabsorption of water back to the blood system. And in this case now, more water will be lost through urine. And the water will be, the urine now will be large quantities and dilute. Okay. The next question
Just a
was...
minute, Emai. Okay, the student was talking about the hypothalamus stimulates the pituitary gland. That gives scientifically it is wrong, yeah? A gland stimulating another gland. So we normally talk of the osmoreceptors of the hypothalamus detects the lower osmotic pressure. Then impulses are generated and transmitted to the pituitary gland that is stimulated to secrete less NDH.
Correct. The function of the hypothalamus part of the brain is to detect. Yeah. Thank you, Hamo. Is to detect the osmotic pressure. Yeah. So after detection, now the impulses are sent to the pituitary gland. So from the information that has been sent from the hypothalamus, the information to do with the impulses to the pituitary gland, it is now, that information now will detect how much antidiuretic hormone can be released. Thank you. Okay. And then the next question was quantity, the effects of the following on the quantity and the composition of urine. Number 2 was drinking of very salty water. So what will be the effect of drinking very salty water on the quantity and the composition of urine? Drinking of very salty water, somebody? What will be its effect on the composition?
Terence?
By drinking
Terence, go ahead.
very salty water can lead to increased urine production as the body tries to get rid of excess salt by increasing the urine output.
Unapotea, Terence. Unable to hear.
I was saying
Mm.
consuming salty water can lead to increased urine production as the body tries to get rid of excess salt by increasing urine output, which helps to maintain.
So drinking a lot of salty water causes production of more urine, concentrated or dilute. Terence. Any other trial? Warren?
I think drinking salty water will increase the osmotic pressure of blood, which is then detected by the osmoreceptors in the hypothalamus, then sends impulses to the pituitary gland, which leads to more secretion of the antidiuretic hormone, which is then carried by the bloodstream to the kidneys and acts on the distal convoluted tubule, hence increasing its permeability to water. Hence, more water is reabsorbed, which leads to secretion of scanty but concentrated urine.
I think that is very correct. It will bring about more reabsorption of water at the distal convoluted tubule. And in this case, this will be done by the antidiuretic hormone. More antidiuretic hormone will be released. And more reabsorption of water back to the blood system. Less water will be released to form urine. Kipruto?
I think impulses are transmitted but not sent.
What's the difference? Impulses are transmitted. The best word description there is transmission, correct? Yes. Now, what about aldosterone hormone? The absorption of salts, what happens? What do you think?
What do you think?
What do you think to do with the aldosterone hormone? Or what do you think about reabsorption of salts? When there is a lot of, when somebody has taken in a lot of salty water. Any trial? Somebody says less sauce will be reabsorbed back to the bloodstream, correct? So of course we expect now that less aldosterone hormone will be released, and therefore less salts will be reabsorbed back to the bloodstream, and more of it now will be excreted through urine. And therefore expect that there'll be highly concentrated urine with salts and less water. The quantities will be lower, but the urine will be highly concentrated with salts. All right. Now, there was a question to do. There's also, he also asked about the quantity and the composition of urine if the pancreas is removed. If the pancreas is removed, what do you think? Faster? Quantity and composition of urine with the removal of pancreas. Somebody had raised his hand na akatoa tena, ni nani huyo? Yes, Kendrick.
If the pancreas is removed, there won't be regulation of glucose in the general circulation. Hence, I think there'll be a high amount of glucose in urine.
Yeah, more glucose will be removed through urine because now there'll be no production of insulin hormone to regulate the amount of glucose. So most of it will be removed through urine. Correct. So that is now will affect the composition of urine. There's another question to do with the. Somebody asked about what? Mellitus. Who was that? Diabetes mellitus. Can somebody remind me of that question? Where was it?
What is the relationship between insulin secretion and reabsorption of glucose at PCT?
What is the relationship between?
Insulin secretion in the pancreas and the glucose reabsorption at the proximal convoluted tubule.
The insulin secretion in the pancreas and the reabsorption of glucose at the PCT.
Yes, so I think it could have been addressed just as the removal of the pancreas. The answer will remain the same.
Yes, it is the same thing because now it has to do. The pancreas has to do with the production of insulin, and if of course less insulin is produced, more glucose is in the blood, and more of it is still released. It's the same thing actually. I think somebody here had asked, please explain how production of less or no insulin results to diabetes mellitus. It might not be related exactly with the expression what we've been explaining here. So, but you can still explain.
Madam Emai,
yes.
The point where you are talking about all glucose is reabsorbed into the bloodstream at the proximal convoluted tubule. That's where it was coming in. So if there is no insulin secreted, then the threshold at the proximal convoluted tubule is surpassed. Therefore, there is less glucose that is going to be reabsorbed at that point. So you find that there is more glucose in the urine. That fella becomes a diabetes mellitus.
That is in case there's no insulin that is produced.
Yes. The threshold at the proximal convoluted tubule is surpassed.
The proximal convoluted tubule will not be able to reabsorb all the glucose.
Yes, correct.
Yes, and therefore some of it will get out.
Excreted in the urine.
Yes, will go through the nephron and be excreted as part of urine. Yeah. All right. I think that is it for today. Thank you, Madam Hamo, for being there with us. And we will meet again next week for another biology lesson. Thank you so much.